Hello friends, welcome back to the course Current Regulatory Requirement for Conducting Clinical Trials in India. Lecture 6, Drug Development Process Overview. So this is one of the important lecture we are going to see. What will we learn here? Drug Development Overview, then Drug Discovery and Developmental Stages, how costly it is to develop a new drug, then why this much time is required for developing new drug, then pathways involved, then what are the preclinical testings required, then what are the clinical testing requires, that is phase 1, 2 and 3, then what is heat, what is lead, all these things we are going to see. About to know in this lecture, as I have mentioned, why it is time consuming, why it is cumbersome, then how difficult it is to have the new drug in the market, then approximately costs involved, then regulatory requirement, what are the forms, what is the fees, and the stages in drug development. So let us start with the introduction. Friends, as you know, in the ancient times, most of the drugs used for the treatment of any disease or disorder were derived from naturally occurring substances of plant origin that we call some Ayurvedic drugs or herbal drugs. For example, quinine derived from a cinchona plant, then opium from the poppy plant, and the other drugs that were derived from the plant extracts. These drugs have been discovered either by identifying the active ingredient from those traditional remedies or by serendipitous discovery. Serendipitous discovery is also called as a happy observation or accidental observation or accidental discovery. But today, majority of these drugs which are used for therapeutic purpose are from synthetic origin. If you see, the most of the drugs, they are from the chemical and synthetic origin, because of having the faster action and some of the other advantages. This is because also the well understanding of the diseases and disease causing factors at molecular level and at atomic level also. Use of technology made it possible to screen huge number of molecules at a time to find its suitability for therapeutic purposes for particular diseases. Even we can screen more than 1 million of the molecule at a time to see whether it is particularly fitting to the target or the protein receptor or not. So the technology has made it possible. Though the technology has developed too much, the cost and time involved to discover and develop a new drug has not been reduced to that much significant level or to that expectation. For example, if we see in UK, it is more than 50 million pounds required to discover and develop a new drug. If we see, every year 20 to 25 molecules only are coming into the market. Why this number is very less? Let us see why and how, where this much cost and this much time is requiring. For this we require to see the drug discovery and development procedure. So this is the flow chart, how the drug is from its laboratory stage, that is the discovery stage, and how it is reaching to the patient, what are the different phases it is crossing through. Here it is mentioned drug discovery, pre-discovery, then drug discovery, then after drug discovery it is a preclinical. So the drug discovery has been made through, nowadays it is most of the time it is based on the computational and that is a STS or CADD, that is computer aided drug design. These softwares are also used for the drug discovery. Once it is found that there is some promising molecules, we call it as a lead or hits, then after its characterization and after finding its suitability, it enters into the preclinical study. In the preclinical study, we can see the animal pharmacology and animal toxicology can be ascertained. and after the successful result of this preclinical study, then one has to apply for the phase 1 clinical trial, that is a new drug or IND application to the CDSCO. Phase 1 generally at least 2 subjects should be there. This is as per our old CDSCO guidelines. However, it is not mentioned in Schedule Y. In Schedule Y it is mentioned that the subject and sites that are mainly depend upon the type of drug for the study and the objective of the study. It is same for the phase 2 and phase 3 also. After completion of the phase 2 and phase 3, the CDSCO review the results, and after having some assurance that drug is safe and effective, then it can be marketed into the market for the patients. This may be some of the time with the phase 4 study condition or without phase 4 study conditions. Let us see how much time is required. So for this drug discovery and preclinical stage, it takes around up to 6 years. To reach these 2 stages, it takes around 3 to 6 years. In the clinical trial stage, it takes around 6 to 7 years, and for the review it takes around 0.5 to 2 years. This may vary from the different sources, little bit there may be variation. So in average we can say it takes around 12 to 15 years for a drug to reach to a patient from its discovery and development stage. From about 5 to 10,000 molecules screened in drug discovery, around 250 molecules enter into the preclinical study, and from these preclinical studies, that is from 250, 5 molecules get entry into the clinical trials, and after having the clinical trial with these 5 molecules, only 1 molecule get approved. So this is the whole process, time required and cost involves. The whole process, it takes investment about $4,800 million over a period of 15 years. This may vary again from the country to country. Let us see the various stages involved in the drug discovery, that is discovery and design. Discovery is a process by which new candidate medications are discovered. So the first procedure while discovering the new drug, that is a target selection. Target selection is the decision to focus on finding an agent with a particular biological action that is anticipated to have therapeutic utility. This decision is influenced by the many of the factors and have to be balanced. This is influenced by scientific, medical and strategic consideration. This target selection can be done by the various computational methods and other methods like SAR, that is structure activity relationship, then HTS, high throughput screening, genomics, proteomics, then chemical synthesis, in vitro study. Sometimes it can be selected through the mode of action of the diseases. Then after the target selection, the process is target identification. Target identification is to identify molecular target that are involved in disease progression, means the factor which is responsible for the disease. That factor have to be identified. The first target has been selected, then the second, the target identification, that is a factor responsible for the disease, has to be identified. These are maybe the proteins or receptors, all this. This can be done by disease mechanism. If we know the disease mechanism, then target can be identified, or disease genes, then target time, durability, functional genomics, all these methods are applied for the target identification. Then after target selection, target identification, it is a time to have the validation of this target. Validation of the target, it means to prove that manipulation of this molecular target can provide therapeutic benefit for the patient. So this is a target validation. After the target validation, the method that is assay development. Assay development is the key factor. It can be in vitro or cell based or using animal, that may be in vivo, or through the STS, that is high throughput screening. So this is to give the assurance about the reliability, robustness of the result produced, to find out the relevance whether that is related to the target or not. Now let us see the drug design, how to identify this target and hits. Identifying the hits. At this stage, lot of compounds are tested against the target proteins. A screening library of lot of number of molecules, that is more than 1 million also, have to cross checked for the identifying the hits, and from these around 1 million compounds, only 100 to 500 hits are observed. Hits are the most promising compounds. So for this method, as I have said, STS, that is high throughput screening, use compound libraries. The STS is the compound libraries are tested against the target proteins, then it is called the high throughput screening. Then once these hits are obtained, then there is a hits to lead identification. It means these hits are further examined or modified to find some promising drug candidate, that is we call as a lead. Means after finding out the hits, again the modification to ascertain again the most promising lead compound. From these hits, 1 to 3 lead compound series are obtained, and once these leads are obtained, then optimization of these leads is required. Optimization of the lead properties, that is with respect to its potency, toxicity, then strength, binding, all these parameters have to be optimized. To identify most promising lead compound, the method like STS or VS are used. VS is the virtual screening method. This can also be used. Actually there are 3 types of virtual screening methods, that is molecular docking, then QSR, and pharmacopoeial mapping. In the drug design process, the other methods like CADD, that is computer aided drug design, then SBDD, that is structure based drug design, these methods are also useful. So after finding out this and optimization of the lead, there is a product characterization. When the candidate molecule shows promise as a therapeutic, it must characterize for its other characteristic, that is molecule size, then shape, strength and weakness, prefer condition for maintaining functional toxicity, bioactive and bioavailability, it must be determined. Characterization studies will undergo analytical method development and analytical validation. So after the characterization, it is a time to have the formulation. So before going any animal studies, the drug has to be formulated. Drug developers must devise a formulation that ensure the proper drug delivery parameters. The drug should be properly delivered. It is critical to begin looking ahead to clinical trial at this phase of the drug development process. Drug formulation and delivery may be refined continuously until and even after the drug's final approval. So this is a continuous procedure. Until it gets the final approval, it has to refine continuously. Scientists determine the drug stability. The stability is also most important, whether the drug sustain the relative humidity, temperature in that formulation, that has to be ascertained. And for all the parameter involved with the storage and shipment, in the transportation, whether that drug will retain its potency and stability, that has to be ascertained. The formulation must remain potent and sterile. See, during transportation and other conditions, it must also remain safe, that is, it should not it should not lose its potency and safety also. It may also be necessary to perform leachable and extractable studies on containers or packing. The drug in which it is packed, that packing material has also to be tested. If it is a solution or syrup or any other liquid preparation, and if the bottles are used, whether that bottles are leaching the material into the content, which may make it more harmful or which may cause it to lose the potency of the drug. So for that, the packaging material study is also required. After having this formulation, the pharmacokinetic and drug disposition study. The studies are primarily in animal models. Pharmacokinetic, that is a PK, and ADME, that is absorption, distribution, metabolism, excretion. These studies provide the useful feedback for formulation. These studies have to be conducted while making the formulation, and it will help in the making the formulation. If we know how it is distributed, how it is going to excrete, then that will help in the formulation. This PK study yield parameters such as AUC, that is area under curve, then Cmax, maximum concentration at which the drug is in blood, Tmax, and the other parameters. Later on, this data from animal PK studies is compared to data from early stage clinical trial to check the predictive power of animal model. The predictive power of animal model has to be checked in the clinical trial phase. Preclinical toxicity testing and IND application. After having this data with the applicants, then preclinical toxicity study has to be conducted. To this preclinical toxicity study, after this preclinical toxicity study, application can be made for the phase 1 as I have mentioned. So during the preclinical stage of the development process, plan for clinical trial and investigational new drug application are prepared. Studies taking place during the preclinical stage should be designed to support the clinical study that will follow. So there are types of studies which is mentioned in the Schedule Y Appendix 3. We have seen in our previous lecture all these appendixes. So what are these studies? Acute toxicity study. Acute toxicity study is to look at the effect of one or more dose administered over a period of up to 24 hours. The goal is to determine toxic dose level and observe clinical indication of toxicity. Usually at least 2 mammalian species are tested here, and data from this acute toxicity studies help to determine the doses for repeated dose in animal and also in human clinical trial, that is a phase 1 studies. The next is for the repeated dose toxicity studies. Depending on the duration of the studies, repeated dose studies may be referred to as subacute or subchronic studies. Sometimes it is also referred as a chronic studies. The specific duration should anticipate the length of the clinical trial that will be conducted on the new drug. Again here 2 species are typically required. Then genetic toxicity studies. These studies assess the likelihood that the drug compound is, whether it is mutagenic or carcinogenic, that has to ascertain in these studies. For this type of studies, procedures such as MS test, that is conducted in a bacteria to detect genetic changes, are used. DNA damage is assessed in some other test. The test is called as a mouse micronucleus test. The chromosomal aberration test and similar procedure to detect damage at the chromosomal level. Then further, the reproductive toxicity test to look at the effect of the drug on fertility, also detect effect to embryonic and postnatal development. In general, reproductive toxicity studies must be completed before a drug can be administered to women of childbearing age. If the drug is of particular nature, then this studies is required. Carcinogenicity studies, these are usually needed only for drugs intended for chronic or recurring conditions. They are time consuming and expensive and must be planned for early in the preclinical testing. Then bioanalytical testing. The bioanalytical testing work is a key to proper characterization of the molecule, assay development, developing optimal method for cell culture or fermentation, determining process yield and providing quality assurance and quality control for the entire development process. It is also critical for supporting preclinical toxicology, pharmacology testing and clinical trial. So these were the toxicity testings. Now let us see the type of preclinical pharmacological testing. After having the toxicity testing, pharmacological, that is safety study, also has to be ascertained in the animals. Specific pharmacological actions are given in the schedule Y, and these specific pharmacological actions are those which demonstrate the therapeutic potential of human. Then some general pharmacological actions, these are also mentioned. These are to study the effect of study drug on vital functions and the vital organs, such as the effect of the drug on cardiovascular system, on central nervous system. All this has to be demonstrated. After completion of these pharmacological general actions, then follow-up and supplemental studies are also required. The follow-up and supplemental safety pharmacology study on CVS and CNS, as we have seen in previous. Here, this study can also be conducted on urinary system, ANS, GI and other organs to investigate the possible adverse pharmacological effect which are not assessed in previous, that is in general pharmacological actions. So other system, that is GI, ANS, these are also have to be tried here and demonstrated. After successful completion of this preclinical stage, that is study in animal, once it is demonstrated that the drug is safe in the animals and there is no toxicity, then it enter into the clinical trial phase. Then one has to apply to the Central Drug Standard Control Organization, that is CLA of India, Central Licensing Authority, for a phase 1. Here it is given the phase 0 also. This is human pharmacology and microdosing. So this phase 0, this is not in India, so it is out of the India. Around 10 to 15 subjects are taken in this study, and it involves dosing a limited number of humans with a limited range of doses for a limited period of time. So before phase 1 in other countries they do the phase 0 study also. The objective of this study is to assess pharmacokinetics, then gather preliminary data on pharmacokinetics, that is PK and bioavailability, to determine if the drug behave as expected from preclinical studies. The study is also called as a microdosing studies. So whether the drug is behaving as expected from the preclinical, this micro study has been done. Then phase 1 study, that is a human pharmacology again. So in our guideline, that is the Indian guideline, at least 2 subjects are required, and in other countries the number of subjects mentioned are 10 to 100. It varies from country to country. So this may involve the first administration to humans, usually to small number of healthy volunteers. So in this study, small number of healthy volunteers are used. Sometimes if the drug is of that nature, then patients also use. Single ascending dose and multiple ascending dose has been studied. Phase 1A, phase 1B. So in India it is only phase 1 we consider. The objective of this study is safety and tolerance. Define or describe pharmacokinetics and pharmacodynamics, determine the dosing, explore drug metabolism and drug interaction, to identify preferred route of administration. So these are the objective of phase 1 studies. Now after getting the successful result of the phase 1 study, then it has to enter into the phase 2 study. The applicant has to submit the phase 1 data to the regulatory authority, and after review of the phase 1 data, the licensing authority may grant permission for the phase 2 study. The phase 2 study is also called as a therapeutic exploratory study. The number of subjects involved at least 10 to 12, in other countries it is 100 to 300, may be undertaken in a larger group of human patients. So here more than the what we have seen in phase 1 studies, there are the small population. Here the population size has to little bit increase with the confidence of the drug having the safety. So the objective of this phase 2 study is efficacy and safety. Here also it is given phase 2A and phase 2B. So in India it is only phase 2 we consider. So phase 2A demonstrate clinical efficacy or biological activity through pilot studies, explore the therapeutic dose range, and phase 2 to determine optimum therapeutic dose and regimen. So here we determine the therapeutic dose and also resolve uncertainties regarding the design and conduct of subsequent trials. After the completion of this phase, then it is a time for the phase 3 study. Phase 3 study, these are the therapeutic confirmatory studies. Means here the therapeutic indication has to be confirmed in these studies. So at least 100 subject, as I have mentioned, it is in our Indian guidelines, and in other countries it may vary from 300 to 3000. This study usually involve a large group of patients. So the number of patients and number of the subjects we have seen from phase 1 to phase 3, it is continuously increasing as we know the drug's safety. The objective of this study, it is clear from the its name of the phase, that is therapeutic confirmatory. So to determine the therapeutic effect in patient for which the drug is intended. If any particular indication is mentioned or proposed, then here that has to be confirmed. It provides a definitive assessment of risk benefit. So in this stage we can ascertain whether the drug is having more benefit than the risk or not. Then increase patient exposure and support marketing claim. So after this phase, one can apply for the marketing permission. Once it has been completed successfully and the result are there with the positive indication, once it is confirmed that the drug can be used in the proposed indication, then the licensing authority may grant approval with or without condition of the phase 4. Let us see what is the phase 4 study. Phase 4, these are after the drug has been marketed. So this also called as post marketing study. The number of patient is not mentioned in our guideline because it is a wide range study and many thousands of the subject patients are involved in these studies. So as I have mentioned, it is a post marketing surveillance study. So the objective to monitor safety in real world. So till now we have seen all in the limited population, now it is in the real world. To refine knowledge of the risk benefit balance. So we have taken the limited subjects in the phase 1, 2 and 3 subjects. Now it is in a wider range. Sometimes describe phase 4 trial as combinations with existing product or to detect rare or long term adverse effect. We have seen that phase 3, phase 2, phase 1, these are for the limited time of duration. Within that period, the sponsor or the CRO, they monitor the patients to see its adverse effect. Now the phase 4 in which the longer duration is taken. So this is about the all phases involved in the clinical trials and drug discovery development. Let us see what is the future direction. So the future direction involves the current approach in drug development is focused on targeting specific signaling pathways. Then despite the new targets such as receptor proteins, then receptor like tyrosine kinase, tumor necrosis factor. Even though all these targets have been identified, still these are not that much effective, and the ineffective therapies with a serious side effects are available. Hundreds of genes could be disrupted in different cancer and in other diseases. Then multiple molecular players and signaling network in diseases, that is also probable reason. So it need better understanding of again the drug target and long-term safety outcomes. The target for drug development and the challenges are the unmet medical needs. In the country and all over, again there is unmet need for the lot of drug for the diseases like oncology, angiogenesis, cell signaling, receptor and molecules and tumor growth. Then cardiovascular and metabolic diseases, type 2 diabetes, obesity, atherosclerosis, thrombosis. Then central nervous system related some diseases like Alzheimer's disease, Parkinson disease, HIV, AIDS is the genuine problem. Novel target in viral life cycle is required. Then some infectious diseases like hepatitis B and hepatitis C, influenza, asthma, COPD. Then some rare diseases and autoimmune and inflammatory diseases like arthritis, psoriasis, inflammatory bowel disease. These are the target for drug development, and these are the unmet medical need. So this is all about the drug discovery, the phases involved and the challenges ahead. So let us see the questions, that is to test your knowledge. The first question is, what is HTS? It is mentioned in one of the slides. So HTS is a high throughput screening. We have seen it is a compound library to be tested with the target protein through high throughput screening. What is a CADD? CADD is a computer aided drug design. So this is a computational. The next question is mouse micronucleus test used to detect. You have to tell what for this test is used in preclinical. So this test is to assist DNA damage in preclinical genetic toxicity study. The next question, that is question 4. Phase 2 clinical trial study is also known as. What is the phase 2 clinical trial study is also called as? So this is also called as therapeutic exploratory study. The similar question, that is a question 5, what is phase 4 clinical study is also known as? So it is a post marketing study, that is after marketing the product in the market. So let us summarize what we have learned in the lecture 6, that is a drug discovery and its overview. So we briefly learned about the stages involved in drug discovery and development. We came to know the time required, it is around 12 to 15 years it takes, and the cost involved, that is around 4,800 million. We also learn about the regulatory pathways, then requirement to bring new drug from lab to the market. We also learn about what is hit, what is lead, what is the optimization of the lead compounds, what are the preclinical stages, what are the clinical stages, the number of subjects required. So this all we have seen in this lecture. So let us wait for the next lecture. We will see in the next lecture. Till then, bye bye, thank you and all the best.